We have Edmund Miller here talking about the state of machine learning for biological data. So thank you very much. Start.
Okay, good morning, everybody. So a little bit about me. I'm a PhD candidate at the University of Texas at Dallas. I'm also an NF Core maintainer, if you're familiar with those pipelines. If not, go check them out. They're great. Lots of great reproducibility being done there. So I've been machine learning curious since about 2017, is what I'd say, and I kind of never really picked it up. I got caught in a lot of the reproducibility of bioinformatics and whatnot, rather than actually applying machine learning, because I also think that there's lots of issues with machine learning and reproducibility. I also never took linear algebra. That's a kind of inhibiting factor as well. So I had some questions to answer about this and kind of figure out about the various loading of biological data and how that would look in Julia compared to some Python projects. And then kind of like what are all the trade-offs of these various packages that are out there and how can we use them in biological data? So, and I also have a personal interest in genomics as well. So, this talk. I have a friend on the Do Emacs channel named Michael, and he used to have this on his GitHub profile of be the PR you want to see in the repo. So I'm going to do the talk that I wanted to see at JuliaCon. So kind of the goal of most biologists is that we're not trying to create novel machine learning models. Some people may try to, but I'm not. I'm just trying to apply these in novel ways to biological systems and biological data, and then make biological inferences using those machine learning models. So I kind of had an old overview and kind of an outline that I was going to go through of kind of calling Python in R packages, what are these different ML packages, and then kind of like loading the biological data formats, et cetera, and some pretty basic toy examples of that. And then I had a thought. What if I reproduce some analyses and then recount what I learned along the way? So what I did was a paper that I was on recently called MLF Core, spun out from some people in NF Core, where we tried to build a reproducible set of tools for machine learning as well, and specifically in biological sciences. And so it kind of goes through various things, and we have models that we support, templating so that you can keep it up to date with the latest best practices without doing a ton of extra work, visualizations. It hooks into ML Flow as well. If you're interested in that, we can always talk about that. It hasn't caught on yet because they're not very passionate about the reproducibility of machine learning. But basically, the entire premise is then using deterministic models rather than non-deterministic models. So I was not responsible for the machine learning aspect of this. I wrote the Python package and did all the templating and whatnot because I was familiar with that from NF Core. So kind of the whole premise of MLF Core was to take ML Flow, build docs with read the docs, support Conda and Docker on those, and create some various tooling. So then that runs in all these different container aspects on the cloud, on HPC, and then all of your runs are reproducible across those so other people can go out and reproduce your research eventually. And so to just get started with that, it was just very simple to do pip install MLF Core, and MLF Core help would then list out the entirety of the tooling. You can list out the various templates like TensorFlow and PyTorch and XGBoost. And then lastly, you can just create those pipelines from that. So why do we care about reproducibility in science so much? As compared to like selling ads or chatbots, which can be variable and there can be some like loss in that. We have lives at stake with these of people getting diagnoses and then scientists who are trying to reproduce this research later down the line. So the first example that we used was called LCEP. And so kind of what it was trying to do was it was trying to classify cancerous liver samples from gene expression data. That's just RNA-seq data for anyone that's familiar with that. It's just basically where you take a bunch of cells and then you sequence the RNA from those cells and then you count them up and then you do some statistical modeling around that. So it was a nice little warm-up. The purpose is to kind of demonstrate the creating of a Python package using MLF Core in the paper and then using that package in a Nextflow pipeline. There's a link to the repo as well, and I'll have a link to the slides at the end. So what it kind of ended up looking like in Python was this data loading section of this. And basically you can see that there's a lot of like conversion to NumPy arrays and then converting from NumPy arrays to XGBoost data matrices as well. And then also going down and then loading this data set and passing all of those out. And then we'll come back to the whole parsing of the TPM table as well, and that's a whole nightmare. Whereas in Julia, it was very simple of I just fed it some URLs, loaded it into a CSV and into a data frame. You also don't have to dance around with, I keep switching between XGBoost and XDBoost. So the data matrices are just automatically loaded from a data frame package as well, and then the wrapper just handles that for you. So you don't have to dance around with NumPy arrays and learn all these various libraries. So this is kind of what the data looks like when it's loaded up. So you have a gene ID over there on the left. The gene name was missing from the data set. And then you have these sample names over here in these columns. And the zeros are for normal data sets, and the ones are for cancerous data sets. And so then this is part of the MLF core example that we had with the Python. And to be fair, pandas probably could have been used here. But it created this entire like elaborate way of parsing this TSV and splitting off the info and all these various things that was difficult to use. And then basically just zipping it up and then sending it back out to do this. But basically so that it could be loaded into a NumPy array was what the purpose of this was, so that it could be sped up. Whereas with Julia, it was very simple, as you saw before. And then we can just go into cleaning this data instead, where we can find all the zeros, for example, and then we can drop the gene name column, and then we can flip the data frame around using some of the functions, and then we can just find which ones are cancerous and which ones are normal and create some labels for this training set. So onto the training of this. There's not much difference between Julia and Python here. Basically, again, you can see the NumPy round, whereas you can just use like just plain Julia all the way through, which is really nice to just be able to follow that in that sense. And I think that kind of touches on the purpose of this and the difficulty with biologists who may not have a background in programming and may get lost in some of these extra things when applying these machine learning libraries. And then when those libraries are then applied, the difficulties with the code reading and other people following along what was happening. So I also wanted to throw some GPUs at it as well. I used a Julia notebook template to using Colab as well for free GPUs, if anyone's interested in that. Basically, again, very simple. It just uses is GPU, and then it hooks up to CUDA.jl, and that just automatically loads the CUDA toolkit for you. So it's just great. It's really simple to use as compared to all of the different pain points that you have with Python and whatnot. So onto the single cell autoencoder. So this is a very common data set. It's 3,000 peripheral blood mononuclear cells is what it is. And it's from 10x Genomics. So single cell RNA sequencing, if you're not familiar with, is RNA sequencing, but then we take each individual cell, give it a barcode, and then we sequence it, and then down the line. So you can actually get various cell types in between that data set, rather than this kind of homogeneous RNA sample from this. So I had an issue with this one, though. Instead of giving a nice TSV, they started with an H5AD data set, which is part of ScanPy. And so they also use ScanPy then to load and clean the data, which is a Python package. So I started trying to reproduce the exact replicate of the ScanPy functionality in Julia. They actually happen to have a Julia package that they've been working on in single cell verse, which is the ScanPy authors. So essentially, the package here should just load up this H5AD file. However, it didn't. It threw an error because it was out of date for whatever reason with the Julia version that I was using. So I got to experience an interesting piece of Julia, is that you can just clone a repo there with dev, and you can just use develop and -- local, and then you can load up this package, of course. Good. So anyways, then that repo is then cloned at dev /muon, And then it's also added to the project.toml for tracking and reproducibility of that, which is also great. And then you can start to just use your entire fork and push that code up and then make a quick PR whenever you finish it and figure out what the bug was. So to be fair, you can also do this in Python with pip-e, but here it's Julia all the way down was the difference. So I could actually hop into the code, mess around with it, and figure it out versus like jumping into some weird C functions and various bindings. So I think that that's really powerful as well when you start to hit errors in the stack in these like packages that no one else is using, for example. So that's above my pay grade to fix that, however. So I then went back and revisited just actually loading a Python package and with this in Python call. So there's then the using conda package, Julia package. And so then all you need to do is just add the conda channel, which is very simple, and then conda add scanpy. And what I love is the toml that it spits out as well. So then it's just fully reproducible from the get-go. Just automatically gets generated in your environment from that. If you haven't seen that, take a look at it as well, compared to some bioinformatics code that I've come across. So I did kind of stumble on that as well. I got confused between whether Python and PyCall were the same package, which is a common thing that new people stumble upon. So I just wanted to include that in the talk as well. So Python call doesn't have as much legacy to support in their section is on this, whereas PyCall doesn't reference Python call, which I think is part of the issue. So Python call support only has to support Julia 1.6 and Python 3.7. So it doesn't have to do as much work as the others. And then whereas PyCall is trying to support 0.7 and 2.7 and onwards. So that's why they can do more in Python call and use things like conda package. And then also using that by default just makes it sane for people to use. So the other added trade-off is that you can use them both at the same time for any reason that you might need that as well. So you're not really having to make a trade-off and make a decision on that. So what this ends up looking like, which I thought was really cool, is you just load this in, you do py import, scanpy, and then I just took the old pre-processing function and just swapped it in, and it just works, which I think is really powerful considering all these other libraries that are pre-written. So this is just going through, normalizing and scaling the data, and then picking out some of the very highly variable genes on those, scaling, and then setting it to that. And so what the old function used to do is it would do some fancy TensorFlow loading and prepare for the model, whereas in Julia, you can just use a natural array and just make it a float 32. And then you'll notice down there that the return of like returning X and then X, because what we're trying to do in this model is go from like plain out to, or like the input to the direct output as well. And we'll talk about that in a moment with the model. So this was the original code, was this whole tensor slices, and then I was trying to dive in and figure out what this is doing exactly. And then I realized, oh, I can just return it in Julia. You don't have to learn what that means specifically and all this various slicing. So I thought that was pretty cool. So I ran into a slight issue where conda doesn't work well on NixOS. So I was running this on our school cluster instead. So I just kind of exported the X matrix so I could work faster rather than rerunning that beginning of the analysis every time. And so then I, of course, had to give a shout out to Data Toolkit, and we'll talk about that for a moment. So what this package is doing is creating an entire data catalog in your repo for your data. So essentially you add the package to your repo there. You can call curly braces, and that switches over to a data REPL. You can then init your data.toml, and then you can add a data set. So I just added that, threw it up on Hugging Face as somewhere to just toss a file and toss the matrix up there. So what would actually be better practice is to use the built-in Julia loader for Data Toolkit as well, and then I could have the entire pre-processing script, then have this second script of like, okay, now I have a matrix here, and then loading from there. But this was just to get it done on that. So what this ends up spitting out is this nice toml here, where you have this description of pre-processed PMBC 3K single-cell data set, on that. And then you can see the driver where it's pulling from the web and the URL, and then the loader, which in this case is just the delimiter library. And then it's just saying that that's just a tab, and then what data type to use for that. And so this is really powerful. You can use this as CSVs, images, all kinds of stuff. And what ends up happening in your code, rather than all that extra loading, is you just get this. And you just can load your data set with the D macro on that and just specify your data set. And so that's really powerful to be able to hand it to another collaborator. I don't know how often y'all have had this, but also often people have given me analyses and then it's like, okay, where'd you get the data from? And they're like, oh, well, I have it locally here, but they can't tell you how they found it on the internet. So if you're interested in that, there's a talk on Friday with Timothy. That's the information for it on the entirety of Data Toolkit. We'll go very into the weeds on it, I promise. So I got a little bit into Flux as well, which is important. It is very elegant. I wanted to emphasize that. I understand why they italicize that afterwards. It just makes a lot more sense than other machine learning libraries, I think. So back to the model of single cell autoencoder. So all that we were trying to do here, and this is very common to do in single cell analyses, is we're just trying to go from input to output the exact same. And what this was trying to demonstrate in the paper is that using these non-deterministic operations, you can end up with not going one-to-one on these. And so that creates a lot of noise in that. And so basically, all of these like various uses of denoising the single cell data and predicting perturbation responses is kind of, is it really happening or is it just part of the randomness of the model? So this was just the Flux loader. So the TensorFlow aspect had a lot of chaining and whatnot, whereas this is very simple just to load both of those data sets, which is the actual training data and the labels there. And then you can just throw in the batch size. And of course, there's more params that you can toss in there as well. And then I pulled this from a blog post on autoencoders on this. So you can just set the device that it's going to be run on, which is the CPU in this case, because it's a relatively small model. And then you can see the various layer sizes of those and specifying those and the batch size for that. And so then this is just the model. And I just thought that it was very elegant and made a lot of sense from someone who's not an expert in this. So this is just taking the size of the data frame, D, up there, and then you're taking the layer 1 from our previous variables here, in case anyone's forgotten that. And then we're just kind of, we're building the model and linking it together of L1 to L2, L2 to L3, and then so forth, and then back out. And then you just have this chaining function down here at the end, and it just loads up by that device. So I just thought this was really easy to understand as a biologist versus all, like you understand the concept and what the model looks like, but then the building of it gets hairy when you start to use these other machine learning models. And so then the training of this, so this is the kind of the for loop way of doing it in Flux as well by the default. And you can see that we've just used the same loss function as the previous one. It was all there, everything that I needed. And then you can see that we just update the model at the very end after we run through and do these gradient responses of that. So this is the fancy Flux train as well. You can also give it a with progress to get a nice taskbar as well on that. And so then we have, you just feed in the model, the training set, and then it just runs this loss function every single time is what it's doing under the hood. And it's much simpler and more condensed to run it that way as well. But they start you out with both in the tutorial. So let's talk a little bit about the liver CT segmentation aspect of this. So the liver tumor segmentation of the computed tomography scans. So just basically it took CT scans. This is an open data set that's out there as well that you can go and download using a UNet model. And then the data in this challenge contains these CT scans, and then the contrast is for various liver lesions. So you can see the liver class over there and then the tumor class on those, and then you can overlap them is what ends up happening. So then the model in this one is a 3D UNet architecture. And so I'm going to be honest, I don't understand how it works, but this is the model that people use for a 3D UNet of that. And so from that, then we needed to load the data, which is the NII files. And of course, there's a Julia package already for that. It's also been, it was 9 years old, which I was kind of surprised by on those. And then the UNet for that is also just already a package as well that's written in Flux. And so then it just has the various convolutions going down and then back up as well on those. I sadly had to cut going through this whole one for time based on those, of not being able to reproduce the entirety of this one. So, in conclusion, some things that we touched on, the simplicity of data loading, I think that that's crucial. The various hacking of dependencies live when you run into a package that's no longer maintained or just out of date. We also talked about calling Python packages from Julia. And then we also talked about Data Toolkit a little bit and how we can have more reproducible analyses with that. And then we also talked about Flux for a bit. And then also we talked about how there were plenty of packages that are already out there ready for biology and Julia on that. So some things I didn't get answers to that kind of my idea of reproducing the ML core that I got distracted from is I'd also like to understand how to load up various BAMs like deep variant and what does that kind of look like on those. So what deep variant does is it takes its alignment from a BAM and a FASTA reference, creates these various examples, and then it creates a convolutional neural net and calls these using that that pile up down there at the bottom and basically running through those. So that's something to stay tuned for that you'll see in a follow-up of this. And then it spits out various variant calls from that. And then I also kind of want to see what Selene would look like as well from the function lab. And so that's just a deep learning library as well for finding various motifs in genetic analyses. And then one thing that I also found missing as well in some of the documentation was how do you have more determinism in these? We talked about all these deterministic models. And I think that's going to be Flux.jl. If anyone has any ideas, feel free to reach out on that. And so links to slides and links to my link tree. Any questions?
I was trying to sense that when we're talking about biological problems, interpretation of machine learning arises as a very important thing because ultimately we do want to be informed about where the species or the genes or the molecules that actually contribute to the phenomenon we're modeling so that they can become like biomarkers or potentially like initial guesses for causal modeling and other things. So I was wondering if you could, if you can, and if you would comment a little bit on the current state for machine learning interpretation in Julia.
For machine learning interpretation.
Because I know that like for tree models, we have very natural invertence measures, for like general linear models, we have like sizes and p-values, we have shapely values, which are agnostic, but like from your entire experience, throughout the story that you told, like what have you learned about interpreting and acquiring biological insight about the stuff that you're modeling?
So I think that's kind of an issue with the various examples. Like this one might be more biological modeling, whereas the first model is kind of binary of like cancerous, not cancerous. So you're not really doing much interpretation there. You're just kind of like, is it cancerous or not? And then in the single cell autoencoder, you're just looking for, can we go from this input of this tensor to that tensor, essentially on that in a reproducible way. So this is more of the modeling that I think that you're looking at, and then the kind of inference from that. Am I following your question?
Yeah, perhaps, but like I disagree that we just want good benchmarks. I think that we like, for example, in this problem, we want to be able to look at the 3D reconstruction of a liver image and say, well, those regions are important, so maybe our MRI technicians have to take good care of them, our image compression techniques have to lose less information on those regions. And this is where, like, for example, UNets are very good at telling us for each channel what are the most important parts of the image, for example. And this is, for biologists, I want to believe, and this is of course a personal opinion, but I want to believe that knowing those things is as important as having accurate predictions. So that's why I'm asking you, like, what do you understand is the current state of interpreting and assigning importance to things we know biologically towards a prediction?
I don't know, it sounds like you got an answer to that. You have an idea.
Actually, as I said, aside from Shapley values, which are agnostic, I don't, and that's why I'm asking that. Because I'm really at a loss right now. If anyone has a perspective.
Well, I just like follow up is like for the single cell data, like just compressing and uncompressing single cell data, like by itself is not valuable, right? What we want to know is how can we understand what that single cell data tells us about the underlying biological phenomena. And having a compressed version, we do something with that we can't do with the giant single cell data that we fed in, right? Like compression and decompression on its own is not super useful unless we're using the compressed data in a novel way.
Right, and then applying that data set to something. Right. So that's kind of more downstream of like various other libraries and making those inferences from that. And I believe there is a talk on single cell analysis in Julia later this week as well. It'll probably be of interest in that case. So that's kind of, yeah, exactly. That would kind of be where it is lacking in that sense. But it's also kind of a discoverability issue with BioJulia that I've found as well. So.
I see that you use mixOS, right? You mentioned that content doesn't work on mixOS. Well, and I agree, it doesn't work very well. But I think there is a way to be, no, I think no, there is certainly a way to build a FHS environment, development environment. In that way, you can use binaries that are not made from mixOS.
Oh yeah, yeah. You can do the whole Mamba, micro Mamba thing in
Yeah,
the
but it's a
shell.
mess. I agree.
Exactly.
But it works.
Exactly.
Yeah, but I see that your presentation you branch off to talk about a lot of stuff, and in the end you talk about biology specifically. But now that you come from Python and you introduce yourself to Julia right now, what do you see are the main differences in your usage, not necessarily performance and stuff, but usage, about TensorFlow and Flux, because I see that you try to transition from one to the other, right?
Mm-hmm.
What is your experience so far?
Exactly, that's a great question. I think that was kind of touching upon the overall theme of I kind of wanted to show it's really simple in that sense, and it really makes a lot of sense. And I think that that's the valuable part of Julia as compared to other programming languages is the scientist's time is the most valuable aspect of that, not really even talking about how fast and compute those are. It's about understanding the concept and then being able to be sure of your computation, not like, well, like having this comment of like, I don't know how this works, but it does, you know? You see a lot of those.
All right. Thank you very much.